Hello, everybody. You are listening to this recording of the chapter 17 and the beginning of chapter 18 lecture. We have a snow day today on Wednesday, and according to the instructions from the administration, we're not supposed to have Zoom sessions. So here I am recording this lecture instead of delivering it live. So I'll be screen sharing lecture slides, and let's take a look at what else we have to talk about in chapter 17. If you remember, we have looked at already three special senses related to mechanoreceptors, special sense of hearing, static and dynamic equilibrium. We started to look at special sense of vision, and we will continue and complete our look at special sense of vision. Chapter 17 has a very short segment on special sense of smell and taste. And after that, we are looking at a few short slides or a few short set of information from the endocrine system as well. The lecture on the endocrine system will be completed on Monday, and hopefully we will have open campus by then. So maybe one more note here, because you are obviously missing a lab today. I will be attaching a recording of that to your class shell. So, and of course, sending copious amounts of messages to keep everybody informed. Let's go back to chapter 17, special sense of vision. And let me see if I can get this screen sharing working. So hope you can all see this slide, and let me see if I can make this full screen. Let me click once again. Right, so here we have a full screen image of the lecture slides. I believe we have looked at these parts of the vascular tunic, and for the gross anatomy point of view, you will see very similar set of information compared to the lab that you already had on the special sense of vision. So in the lab, we mentioned all this for the vascular tunic, the choroid, the ciliary body, and the iris. And I believe we have looked at both of these components in our previous lecture. What we haven't looked at in detail, and we have a little additional information here about the iris. So from the outside, the iris is this colored portion of the eye. And this colored circular ribbon encloses an opening, the pupil. We need this pupil because light is penetrating through the pupil deeper into the eyeball. Because the iris has smooth muscles, it is strategically located to regulate the amount of the light that enters the eye. Take a look at this pupil here. It makes sense. The wider the pupil, the more light will enter the eye. So narrowing or dilation of the pupil is brought about by the smooth muscle action of the iris, and it regulates light entering the eye. This is the view that we didn't have in the lab. We're looking at the iris from the backside, and if we look at this set of muscles, we have a circular and the radial smooth muscles. Under normal light conditions, average light exposure, we have an intermediate diameter to the pupil. Think about the situation when you step into bright sunlight, or you can do this artificially, turn on the flashlight on your phone and hold it up to your eyes. The intense light will create this reflex where the circular smooth muscles will contract and narrow the lumen, narrow the pupil. So this constriction of pupil is brought about by changing light conditions. On the flip side, when you step into a poorly lit area, or you are in fight or flight response mode, it's the radial smooth muscles that will contract, and those muscles will stretch the pupil into a much larger diameter. So this is the dilation of the pupil. What you see here in parentheses is the division of our autonomic nervous system that brings about this contraction. So activation of the sympathetic division of our nervous system means that we are in an alert mode. We are scared or frightened or intimidated by something or somebody, and these nerve impulses will automatically make our pupil dilate in fight or flight response mode. When we are dominated by nerve impulses from the parasympathetic division, rest and digest conditions of the body, our pupil can be narrowed if needed for constriction. So this is the way those two smooth muscles act to regulate the amount of light, either constricting or dilating the pupil. We mentioned this in the lab as well, that while it's near the vascular tunic, the lens is not really part of the vascular tunic. It has no blood vessels. The lens is made transparent by proteins, very different way than the cornea becomes transparent. Nonetheless, the lens is yet another transparent tissue. It has the property of refraction, or the ability of the focus light, and it is located right behind the iris in our eyeball. A closer look at the lens reveals that the lens is attached to the ciliary body through these suspensory ligaments. We will see another illustration of the change in the shape of the lens accommodation. So what you see here is a situation where the suspensory ligaments are tight, they're straight, and they are stretching the lens into this flat shape. This person is looking at objects further than 20 feet. Ciliary muscle is relaxed in this condition. If we are looking at an object closer than 20 feet, the ciliary muscles will contract and they will relax the suspensory ligaments, allowing the lens to take a more curved, a more chubby, a shorter shape. And now we can focus light from objects closer than 20 feet. This is also a good view of the lens and how light is coming through it. There's a pathology to the lens. When the lens becomes cloudy, obviously it will not allow light into the retina, into the rest of the eye very efficiently. So that takes away from our visual experience. Excuse me. And that condition, the cloudiness of the lens, is called cataract. To fix a cloudy lens, especially in an aging population where this is more and more common, we do have what's called cataract surgery. So cataract surgery is made very easy because the lens is included in this pouch called the capsule. And during cataract surgery, the capsule is slit open. The old lens is crushed inside a capsule with ultrasound or laser light, and the old broken up lens is suctioned out. and a new plastic or glass lens is slipped into the capsule to fill the place of the old one. So that is cataract surgery, replacing an aging cloudy lens with a plastic or glass replacement. This is how important for us the lens to be transparent and being able to focus the light. In the lab, you will be seeing you have already seen a lab related to the vision. So we had a monkey eye slide in that lab, and that's where we explained the location of the aqueous humor and vitreous humor. So in the posterior cavity, let's start with that. On that cow eye dissection, you have seen this jelly-like clear mass coming out from the cut open eyeball. That was vitreous humor occupying the posterior cavity. The other type of liquid, the aqueous humor, is filling the anterior cavity. And we have a little additional segment here on the aqueous humor. It is the ciliary process that makes aqueous humor. This aqueous humor is produced constantly. The freshly produced aqueous humor moves into the anterior cavity all the way up to the neighborhood of the cornea. And this fluid is constantly drained away by this venous sinus. A disorder of the circulation of the aqueous humor is when this drainage is plugged or somehow narrowed. That condition builds up the aqueous humor and pressure in the anterior cavity. That condition is called glaucoma. A glaucoma is fixed or helped either by drug treatment or by surgical opening up of this drainage. So fluid buildup is prevented that way. The last layer excuse me, I have to blow my nose. The last layer in bright yellow of the three tunics is the nervous tunic or retina. There's a lot of interesting information that can be derived from observing the retina. Take a look at this image here. You can see the retinal blood vessels, the meandering, branching pattern of the blood vessels is unique to each person. When you hear about a personal identification method called the retinal scan, it means a scan of these blood vessels. Again, this is more unique than a fingerprint to every individual. So it's a great way for personal identification. In an optometrist office where the blood vessels can be imaged every visit, later visits may point to a degradation or degeneration of some of these blood vessels. Branches may disappear, and that's a warning sign. In case of high blood pressure, hypertension, or diabetes, after years of those uncontrolled conditions, the smallest blood vessels, the capillaries in our body, among them on the surface of the retina, will be damaged, and the damaged blood vessels will be degraded, or the branching will be cut back. So retinal blood vessels can inform us about about disorders that may not be noticeable by other means at the particular time in the body. We also want to mention this condition, detached retina. The vitreous humor is pushing the retina right on top of the choroid. Retina needs to be maintained in this shape to be in the focal point of light focused by the lens. If a part of the retina is detached, usually a big bubble of liquid forms underneath, and that detached retina is removed from the focal point, leading to blurry peripheral vision. And sometimes the detached retina actually curves under here, and then it creates a totally dark region in our visual field. So the detached retina is most frequently fixed by surgical reattachment of the retina. Minor detachment may heal on their own. Here's the artistic rendition of the layers of the retina. In the vision lab, you had a retinal slide that showed the retina in the reverse position. On that lab slide, the light was coming in from the top. Here, you can take a look at this yellow arrow. It tells you that light is coming from below. So it's like you're looking at something on the floor and light is coming into your eye. Here is the layer for retinal blood vessels. And here are those layers we mentioned in the lab as well: ganglion, bipolar, and the photoreceptor layers. The pigment epithelium plus the choroid absorbs stray light as light is going through here. With the photoreceptors, we're staying still very close to the amount of information we mentioned in the lab. In the lab, we mentioned rods and cones. We mentioned their different sensitivity to light. Rods sees or detects shades of gray. Cones detect color. And it's the cones that are extremely densely packed in the macula lutea. And this macula lutea, therefore, gives us the sharpest resolution, best visual acuity, and also the best color vision. A disease of this region of the retina is called macular degeneration. Macular degeneration causes the degradation of the rods and cones in the macula lutea and takes away this most precious part of the retina. So in terms of responding to light, two parts of this process is described here. We are going to detail both of them. First, when a nerve impulse is created here in response to light, it is the bipolar cells that get excited first. They create the first nerve impulse. Nerve impulse will move onto the ganglion layer, and from the ganglion cell layer, nerve impulse will be taken out through cranial nerve 2, and we'll see a detailed view of that on the next slide. We will also detail, as opposed to not saying anything about this in the lab, we're going to detail the process, what is happening in rods and cones, how is this light can be transformed into change in the receptor cell, and we'll be seeing the role of this neurotransmitter glutamate in this process. So let's start with the pathway first. So from a retina, those bipolar and ganglion cells will be relaying nerve impulses to cranial nerve 2. On this image, you can see the cut cranial nerve 2 from both eyes. The two cranial nerves cross at this optic chiasm, and then they split again into these two optic tracts. The optic tracts take nerve impulse to the thalamus, and from the thalamus, nerve impulses are taken to the occipital lobe in the cerebral cortex. And once the nerve impulse reaches the occipital lobe, we do have conscious awareness of light in our environment. So this is the pathway of nerve impulses. So let's get back to what's happening inside the rods and cones. Here is a magnified view of a cone, and right here, magnified view of a rod. Light must react with something to cause a chemical change inside these photoreceptors. The agents that will change chemically are photopigments, and photopigments are stashed into these discs of the cones and of the rods. For the rods, the opsin portion, the protein portion of the photopigment is called rhodopsin. For the cones, the opsin has a green, red, and blue version of that opsin protein. Retinol, this small lipid, is an essential part of all photopigments, whether in rods or cones, whether it's rhodopsin or red or green or blue opsin. So when we state that the cone contains 3 different kinds of photopigments, so it means red, green, or blue opsin. Opsins are proteins, and the DNA that encodes these opsin proteins can undergo mutation. Color blindness is the mutation in those opsin proteins, and those mutations disable either red or green or blue cones. I'm sorry, I'm going to have to move through here. So physiology of vision includes the processing of information. And I apologize, it looks like I'm missing the slide here. Let me go and see if I can find. Nope. So let me talk about that slide, and I'm not sure how that is. Nonetheless, in your class, the sequence of slides includes an additional slides that explains how rods and cones react or behave in dark versus light. And I'm going to go back here. This glutamate is released in abundance in dark, inhibiting the bipolar cells, and the slowing of glutamate release will be leading to the excitation of the bipolar cell. So that's the tremendous difference between dark and light. The receptors, rods and cones, will slow this inhibitory neurotransmitter release to lead to the excitation of the bipolar cells. Processing of visual information. This is mostly expressed by this portion here. Even inside the retina, nerve impulses are extinguished or enhanced by the horizontal or amacrine cells. So it's not only brain centers that process visual information, even retina and these 2 cells can enhance contrast, therefore improving our visual experience. The convergence of cones explains the sensitivity of these cones to very, very dim light. The reason why we can still detect and gain an image in dim light, because hundreds of rods are connected to a single bipolar cell, and you can see a bunch of rods and their connection to this bipolar cell. So even if a single rod is excited out of that 600, that will create a nerve impulse by the bipolar cell. So very low level of lights can be created by this network, by the convergence of the rods in the retina. In terms of image formation, let's say a few words about this mysterious refraction. We mentioned this in the lab. And here you see how the light rays change direction. This change in the direction is essential for focusing of the light. So this is why refraction by the cornea or the lens is so important. Direction of light changes once light enters into those tissues, cornea and lens. This is the best image to explain the accommodation of lens changing in the shape of the lens. So when we're looking at an object further than 20 feet away, our lens is in this flat shape. The ciliary muscles are relaxed. When we are looking at an object closer than 20 feet, ciliary muscles will be told to contract, and the relaxation of the suspensory ligaments allow the lens to take a more curved shape. Compare these two shapes of the lens. You can see the shorter and more curvature. This is going to give us a different focusing property of the lens, and objects closer than 20 feet can be now focused on the macula lutea here. And the last topic here in chapter 17 is convergence of the eyes. This convergence is the result of angling of the eyeballs. You know by now from the lab and also from the previous part of this lecture in chapter 17 that it is this group of skeletal muscles in the eye socket that move the eyeball. So it is the coordination of those muscles that create the convergence of eyes. This is pretty useful because we derive the three-dimensional vision and depth perception as a consequence of the convergence of the eyes. So this is the end of this is the end of the visual special sense of vision in chapter 17. If you have any questions, please send me a message. We are going to switch to the chemoreceptor-based special senses here. Now let me switch sharing here. And if everything goes well, this will be the expected section, right? Let's do this sharing. So here are the chemoreceptor-based special senses, special sense of smell and taste. In addition to the cerebral cortex, which we will emphasize as destination, the limbic system, the emotional brain, as some people call it, also receive nerve impulses. So especially detecting familiar or long remembered smell or taste may bring up strong emotional reactions as a consequence of this connection to the emotional brain, the limbic system. The olfactory epithelium, we mentioned the structure in the lab. We do have a supporting role of the ethmoid bone, whether cribriform plate or nasal concave. And when we get close to this olfactory epithelium, we are seeing the blue olfactory receptors. These are neurons. Their axons form cranial nerve 1 as they move through the olfactory foramina. And you see these nerve impulses will be passed on to the olfactory bulb on its way to the destination of the cerebral cortex. You see the odorant molecules. These are the chemicals that are moving around in our nasal cavity. These odorant molecules must run into receptors, receptor molecules on these olfactory hairs. So here is an olfactory hair magnified. This beige portion is the inside of the olfactory hair. On the outside here is an odorant molecule. Let's say this odorant molecule is a methylmercaptan. Methylmercaptan is this chemical that makes gas smell bad. And so this is an odorant molecule moving around in our nasal cavity. For us to have a detection of gas leak, this odorant molecule, this methylmercaptan chemical, must contact physically this olfactory receptor protein. What happens after this contact is a push over several dominoes. G protein will be activated. The active G protein will turn on this adenylate cyclase factory, and cyclic AMP from this factory will open sodium channels. Once sodium channels are open in this neuron, the olfactory receptor cell, beginning of the depolarization is created, and eventually action potential will develop and a nerve impulse will be taken through the axon of this olfactory receptor cell. So this is the set of molecular events that allow us to create a nerve impulse after detecting an odorant molecule. Adaptation is very quick in special sense of smell. Here is the mention of this methylmercaptan, the odorant that's added to natural gas. In the 1930s, there was a major explosion in a Texas high school. That was the major motivation to add this odorant to natural gas. Without this odorant, methylmercaptan, natural gas would have no smell and we could not detect it. The olfactory pathway, of course, starts with the olfactory receptor cells. These are neurons and their axons form cranial nerve 1. Cranial nerve 1 takes nerve impulses to the olfactory bulb. You see this little yellow structure here. And from the olfactory bulb, neurons will go all the way to the temporal lobe. And temporal lobe is the destination for these nerve impulses. Conscious awareness of smell begins once nerve impulses reach the temporal lobe from the olfactory bulb. A special sense of taste, also based on chemoreceptors, we call them taste receptors. In the lab, we mentioned the sour receptors detecting hydrogen ions, the bitter receptors detecting small organic chemicals, sweet receptors detecting simple sugars, salty receptors detecting sodium ions, and umami receptor types detecting amino acids. We also mentioned in the lab that taste receptors are located in taste buds. We're going to see a magnified view of that in a little while, but here in a more bird's eye view, you can see these blue ovals. Those are the taste buds at the bottom of papillae. This is a valate papilla, and you can see the taste buds and the taste receptors are well protected from all the abrasion and physical activity on the top of the surface of the tongue. Here is the taste bud. So the taste receptors are receptor cells. These blue receptor cells will have to create neurotransmitters to excite the sensory neurons, and nerve impulses from these first order neurons will be taken to the brain for detection of the taste sensations. Here is the gustatory pathway. From the surface of the tongue, either cranial nerve 7 or cranial nerve 9 will take those taste-related nerve impulses into the thalamus, and then into the parietal lobe, and we become consciously aware of taste sensations once the nerve impulse arrives in the parietal lobe. So this is the brief description of chemoreceptor-based special senses, special sense of smell, and special sense of taste. This is the end to chapter 17, and we have a little bit of time to look at the beginning of chapter 18. If you look at the schedule, we have this endocrine system opening schedule for this lecture. So let's take a look at this, and we will be continuing the endocrine system on Monday. Let's hope that we have a face-to-face class that time. So let me switch over to the endocrine system slide. And that screen share will bring us to the endocrine system. Let me make this full screen. So the endocrine system is listed right after the nervous system because the endocrine system also controls many activities in our body. We know that the nervous system controls many actions from chapter 15 AMP1. We have seen nervous system controlling heart rate, the activity of sweat glands, controlling blood pressure through acting blood vessels. Nervous system controls the dilation of pupil. We have seen that even in chapter 17. And of course, in chapter 16, we talked about nervous system controlling skeletal muscles. So in addition to those structures, we have many, mostly visceral organs that are controlled by a totally different means. And the control is chemical mediators, chemical instruction carrying molecules called hormones. So the endocrine system is made up of endocrine glands that release hormones into the blood. Hormones control body activities in a much slower fashion. It takes just a second to make and guide nerve impulses to a target organ. It may take hours to make the hormones, release them, and allow the bloodstream to take them to the target. So hormones act slower, but hormones are well situated to provide long-term regulation because hormones stay in the bloodstream for weeks or months. In terms of general functions of hormones, let's just say that many aspects of the body function, homeostasis is controlled by hormones. We'll see examples, hormones controlling the chemistry of extracellular fluid, for example, blood, water, calcium, sodium, potassium levels. Some hormones control metabolism, speed of chemical reaction, and therefore heat production in the body. Some hormones control biological clock. They work together in sync with the hypothalamus. We'll see a very brief example of this. We will be seeing hormones controlling cardiac and smooth muscle action, secretions of other glands, and many more that we will mention the specifics once we get to a particular endocrine gland and their hormones. Nonetheless, hormones are tremendously powerful instruments of maintaining homeostasis in the body. When we define endocrine glands, these are glands that produce chemical mediators, chemical instructions, hormones into the bloodstream. Before we go any deeper into endocrine glands, we have to mention, and this is a review from chapter 4, way back from AMP1, that there are those glands that are called exocrine. A sweat gland is probably a good example because you can see the secretion on the skin surface, but a salivary gland is also exocrine gland. Our liver is an exocrine gland. Secretions are produced onto either exterior or internal body surface. Endocrine glands, they release their products into bloodstream. In terms of producing hormones, pituitary, thyroid, parathyroid, adrenal, pineal glands are full-time endocrine glands. They do nothing else but produce hormones and release them into the blood. There is a surprising number of other organs that release hormones into the blood, but those other organs have functions not related to hormone production as well. Hypothalamus, we have already seen the role of hypothalamus in regulating the switch between fight or flight response versus rest and digest activity in chapter 15. In chapter 16, we've seen the hypothalamus regulate wakefulness versus sleep. Hypothalamus is also a location for hormone production. We'll be seeing that in Monday's lecture. The thymus gland has a major role in our immune system, yet local hormones are produced here. Pancreas is a major gland for our digestive system, yet two very important hormones are made here. And even organs that you may not think make hormones, kidney, heart, placenta, small intestine, stomach, these will be on our menu as we look at urinary digestive system or cardiovascular system. This is the schematics of what's happening in the endocrine system. So in our endocrine glands, there are cells that are making this hormone. You can see the hormone is pushed out of the producing cell and enters the bloodstream. In order to be detected by cells, we have to have hormone receptors on the cells. In a lab, we explain this in a probably simpler fashion. Nonetheless, hormones can target only cells, tissues, organs, if those cells have receptors. The receptors fit the shape of a hormone. Think about a hormone receptor as a baseball mitt. The baseball mitt very comfortably can envelope a baseball. A different hormone, let's say shaped in the shape of a football, will no longer fit into that baseball mitt. So that football-shaped hormone need a different receptor that can accommodate the football shape. And this is how selectively receptors can recognize individual hormones. We were going to mention one example specifically in chapter 18 of local hormones. Local hormones act within a particular organ, stimulating neighboring cells through the paracrine fashion, or sometimes stimulating the producing cell itself. These paracrine and autocrine stimulations have tremendous role in embryonic development. Again, we will explore the role of one local hormone, which will be coming up in Monday's lecture. So how hormones act on the targets really depends on what the message of the hormone. Some hormones instruct the cells to make new molecules. Some hormones instruct the cells to change the permeability of the membrane, allow water, sodium, calcium to move through. And some hormones change the speed of chemical reaction, metabolic reaction in the cells. Hormone receptors can vary in their numbers. This has a lot to do with how sensitive or insensitive a cell to hormones. We're not going to say a whole lot about this. I wanted to mention this change in the hormone receptor number because it tells you that this is an active area of the life of a cell. Cells can push up the number of receptors or take away a lot of receptors from cells. And maybe this is the best place to stop. We'll be getting into the classification of hormones, and we'll explore that in Monday's lab. So I'm going to stop this recording here and stop screen sharing. So this is to replace today's face-to-face lecture, and I'll be attaching this to the class shell with an explanatory class message as well. So thank you for listening. I hope you have a great rest of Wednesday. And of course, a recording of the lab will also be coming to you with a separate message as well. So have a great rest of the week, and I hope to see you, all of you, in person, face to face on Monday.